Today's first speaker is Dr. Boyd Haley. Dr. Haley is a research scientist and international lecturer with a degree in biochemistry and chemistry from Washington State University and has numerous research studies published in peer-reviewed medical scientific journals. Aside from being the former chairman of the Department of Chemistry at the University of Kentucky, his published research includes effects of mercury on neurologic tissue, specifically tubulin, simulating the pathology found in Alzheimer's disease. And not too long ago, he addressed the United States House of Representatives Committee on Government Reform and Oversight. So please give a warm welcome to Dr. Boyd Haley.
As the slides talks about here, we talk about Dr. Michael Ziff, and most of you would remember him. And he was absolutely fundamental in me being a part of this academy. He was the first one to contact me at the request, I think, of Robert Reeves when I first reported mercury causing the same biochemical effects you see in Alzheimer's disease. And Mike was incessant. I mean, I can't tell you how many semi-unpleasant conversations we had on the phone, and some of them were very pleasant. But he never gave up, and I never gave up. And he came up with this mantra, at least this is where I heard it, was this, you know, show me your science, because I told him I wasn't about to get involved with people that were flipping about research and what might cause diseases and just taking wild guesses based on emotion. And there were a lot of things when I first came into this academy. First of all, I didn't believe the FDA would allow somebody to put something in my mouth that would leak micrograms of mercury every day. And we had some severe discussions about this, along with David Kennedy, as I remember. And David's the one that brought me down to the ground with his famous philosophical statement, "Boyd, they lie." And, you know, and I really didn't believe him at that time. But when somebody says something with straight conviction, looking you straight in the eye, you go check them out. And you're right, David, they lie. And so I'd like to dedicate this to Mike Ziff. I really miss him. I mean, he was a good friend, and but Mike wasn't trying necessarily to be my friend as he was trying to get something accomplished, and that's, you know, to help this academy grow and to get the science involved in dentistry, which, as we do the talk today, you'll see that it's improving, but it's still not there. This is something that came out, and I don't know why my thing is showing up, but there's a focal infection theory. When I came into it, this was really a hot topic. And so today I want to start out right away with a disclaimer. I've only looked into one person's mouth intently for more than 3 seconds. I didn't like it, and I've never gone back there. So I'm not a dentist, but I do understand the biochemistry and the microbiology that's associated with this, and I'm going to try and keep my talk with that aspect. But when you read the dental literature, and I do love reading it because it makes a lot of sense, and there's a lot of good research published in dental journals. But they had the thing called the focal infection theory, and it had been disproved according to the American Dental Association and the Organization of Endodontics, etc. And so when we look at this, it's now a fact. You don't hear them saying, oh my God, we were wrong. But when you look at the publications, which we're going to go through a large number of them today, and they're in your handout, there's no doubt that most of the infections that end up in your body affecting certain organs comes from the oral cavity, from periodontal disease or infected teeth. And we talk about this thing, do toxin-producing microbes reside within the dental tubules of vital but periodontally diseased teeth? Are they doing some problems? And then, if so, what about non-vital, endodontically treated teeth? Another question. And so, in other words, are periodontally diseased teeth and endodontically treated teeth infected? We're going to show you that they are. And we're going to show you that the primary research done by dentists today are way off track. They're looking for endotoxins. There are endotoxins, but the toxins that are released that cause the local disruption of the tissues that are infected excuse me, are small molecule toxins. They're reactive thiocompounds. And so we can talk about this, and this is something that will be brought up. Root canals, infected versus toxic, because every root canal tooth, trust me, is infected. Whether it's toxic or not depends upon the nature of the bacteria that's done the infection. And we can talk about whether these are clinical or subclinical. We're going to present data that addresses this. Is it acute or chronic? And does the infection constitute a foci of infection that can become systemic? I think I'm going to give you convincing evidence from other people's laboratories that this is the case. And I would point out to you, the vast majority of this research is not done in the United States. It's not done here. We are practicing third world dentistry at the research level in the United States. We're way behind the people in Finland, Norway, Sweden, Germany, Japan, etc. if you're going to address all this thing, with me as a scientist, I go to the very bottom. And you ask, are extracts prepared from extracted avital teeth toxic to mammalian enzymes in vitro? If this doesn't happen, then you don't have an argument or anything to measure. To get the general overview, this was published in 1994 by Debillion, and he listed several things that were infected. Systemic disease, he proposed, were caused by oral microorganisms. And if you look at them, from heart infections, brain, skin, eye, lung, hematological, implant infections, and jawbone osteonecrosis as mine, I put the question marks by it because I think that's one of the more serious illnesses that come up, and medicine or American dentistry totally ignores that. Matter of fact, they don't ignore it, they tell you that it doesn't happen, and they're totally wrong. And the one thing that we really can address that gets to people right away is the increase and the correlation between periodontal disease and preterm births. This costs the medical establishment or the medical insurance companies in this country, you know, hundreds of millions, if not billions of dollars a year. And it's a direct correlation. The risk of preterm low birth weight babies and every disease that goes along with that for the infants increases with the severity of periodontal disease in the mother. And we're going to show you now the biochemical evidence that shows that the bacteria from the mouth get into the placenta and infect it and cause these problems. And we're going to show you the toxicity that's associated with this. We need to do an evaluation of the vital versus avital teeth, and we can look at the curvicular fluid next to teeth because it should give us some meaningful way of looking, are we helping these patients when we treat their mouths? Can we get the infection in the periodontal cavities down, in the avital teeth down? Can we tell if they're there? And what I'm going to try and present to you is if we identify the proteins and the toxins in the curvicular fluid, we can tell whether or not that site is infected. I can't tell you a lot about the tooth with that test, but what I can tell you is that there are toxins there. Now, I was given a paper to read about, and I immediately laid down, forgot about it. I am associated with Affinity Labeling Technologies, Alt Biosciences it's called, and we have developed a test that I'll talk about here a bit for you to use. So I do have some commercial interest in what's being presented here. But we don't have a booth here today. We decided not to do that because of this thing. Because of the talk, and it would look like I was trying to do it to promote selling something. So you can't buy anything from us today. This is the first one, is occurrence of invading bacteria in radicular dent in the periodontally diseased teeth, then the microbiological findings. Again, if you look at the names here, not done in the United States. And what they did was a simple thing. They pulled teeth from people with periodontal disease and people without periodontal disease, and they stripped off the cementum, and then they took a dentin sample. And what they were trying to find out, were there bacteria in these dentin samples? And to do this, they cultured this sample using an extract of this without crushing the dentin sample, and then after they crushed it, thinking if there were bacteria inside, it would be released, and you'd get more colonies formed. And if you look at the data down here at the bottom, this was the test samples, and this was the level in the infected teeth, which was 501 before they homogenized the sample to break it up, and it was 500 versus 12 in the control samples, the teeth that were pulled with people without periodontal disease. After they crushed them or after they homogenized them, this went from 500 to 3,463, where this one went from 12 to 13. So what we're seeing is that, indeed, if you have your teeth and you have periodontal disease, the teeth are still alive, the bacteria are invading that teeth. They're going into the dentin tubules, and they can go through the cementum, and they don't have to duck when they go through. If you look at the electron microscopy, there's plenty of room for them to get in, and that's how you get an infected tooth and a tooth that, quote, needs a root canal, because you formed an abscess at the bottom, because they come in from the periodontal pocket into the tooth, down to the end, and the body responds to it, trying to fight it off, and it develops this type of infection. And you'll notice everything I have here, I'll have in blue the relevant journal that this was published in. So we take the next step, and this is the ultrastructure observations on bacterial invasion in cementum and radicular dentin. And again, this was done by Walter Loesch. He's a very prominent American researcher at the University of Michigan School of Dentistry, and I think he gets hammered on quite a bit early on. We've had him talk at this academy. He's a very good researcher, but Adrian's and these other folks were from the University of Ghent in Belgium. And this is the electron microscopy, and if you look at that, that's a dentin tubule hole right there, and you see the bacteria invading it, the filamentous bacteria. And if you look here at a bigger picture, you can see the bacteria loading up in the dentin tubules. When I first got into this, I remember reading a paper where they were attacking West and Price, who said that teeth became infected, were invaded by bacteria. And the argument from the opposition was there wasn't enough room in these dentin tubules for bacteria to get into them, and the cementum kept them out. And this is living proof, this paper and several others, that Weston Price was right and the other people were wrong. These teeth do become infected. They carry the infectious material back to the tooth, and you can't get rid of it by any easy means. So if we look at this paper, and you can read this as well as I can, the bacterial invasion of this. And so what they did, and if I can look at this, get you this study, they started on the inside of the tooth. Like there's where the plaque was from the periodontal disease. So they took this part of the tooth and they layered it. And they were looking at the amount of bacteria that would be from the outside to the inside in the pulp area of the tooth. In measuring this, they looked at the colony-stimulating factors of a healthy tooth versus a diseased tooth. The healthy tooth is in blue, and you can see it's 5.4, 7.6, 0, and 2.3. In contrast, going from the pulp side, in the infected tooth, it was 1,399, 2,263, 1,464, and then 16,000 on the outer edge next to the periodontal cavity. So what you can say is that the source, the bacteria, is going from the outside in, getting into the pulp, and it's going to infect this tooth. And so if you have this amount of colonies going from 16,000 down to, say, 1,400, whereas with this one here you're going from 2 to 5, you can say that bacteria are constantly invading the tooth, and it can go from the outside into the pulp, and that seems to be the major direction that it's going. In other words, it's not coming from the pulp out, it's going from the pulp in. So you are having an invasion. And so their net thing was the bacterial growth was detected in 87% of the periodontally diseased teeth. 59% of the teeth contained bacteria in the pulp sample, so indicating that the outside from the periodontal had gone into the pulp, and this was quite a bit. And you can look at the numbers, 259 to 7,000 times greater than the concentration that is the corresponding layers from healthy teeth. And so what we can say is that the radicular structures likely serve as bacterial reservoirs from which bacteria recolonization of the mechanically treated root surfaces can occur. In other words, if you're cleaning a tooth and you're doing root planing and scaling, you're opening up that colony, that periodontal pocket, to bacteria to come out of the tooth back into the periodontal pocket. So you have a problem here. You definitely have to clean up the tooth, but at the same time you're increasing the rate that the bacteria can come back out of the tooth and reinfect that pocket that you've just treated. And so we have to have an intelligent approach to treating this type of situation. So we have, we can address several major questions, and this isn't all of them, but we can say, do bacteria from the periodontal disease pockets enter the bloodstream? And I'm going to show you a publication later where they treat people with heart disease, and they treat them with antibiotics, and these people improve. You take away the antibiotics, the infection comes back. That seems to be a normal situation, and what I would propose to you is, and what these people propose, and something that we said many years ago along with Weston Price, is that you cannot stop the infection in a periodontally infected tooth, or a bacterial infected. You cannot get the antibiotics in there to kill that tooth, that infection. The antibiotics do not get into the dentin tubules and eliminate that infection. And so we can ask other questions in this, do any of these bacteria cause systemic infections and relevant diseases? We're going to discuss this and give several examples of recent publications that indicate that. Do the infected avital and endodontically teeth lead to blood bacteremia, similar as proposed for periodontal disease? And I can't believe they don't, but that will be up to your decision to make that. And we can look at this like this. Do periodontal disease and infected teeth lead to jawbone osteonecrosis? And does jawbone osteonecrosis lead to other systemic illnesses? This is the big argument now with regard to cavitations, etc. So what happened? And this was an area of argument that it seemed to be tested by microbiology and the techniques that most of you know about. But about 20 years ago, new technologies came in, and it's very difficult to detect anaerobic bacteria because they die. You expose them a little bit to air, they don't culture very well. And so you have this problem if you try to show that bacteria from a root canal ended up in your liver. It's very difficult to isolate that and culture it. As a matter of fact, almost impossible. But they had this new technique. It's called the polymerase chain reaction, or PCR. And for those of you that don't follow it, that's when you watch CSI and they get a very small drop of blood from a sample, and then they go back and they say, oh, this DNA came from Joe Smith, and so he's the killer. And what they do is they have a technique where they amplify the nucleic acid, and that nucleic acid will identify either the human subject or the bacterial species that put the DNA there. And so this is what this allows us to do. It allows us to go into a person who has an infected site, pull out that bacteria, even if it dies. We can isolate the DNA, RNA from that bacteria, amplify it, and say, oh, this belongs to this specific species of bacteria. We can then go to their heart tissue, their atherosclerotic plaques, their cardiovascular, or the placenta, etc. We can isolate the bacterial fraction from that, take the DNA, amplify it, and say, oh, the bacteria from the mouth ended up in this lady's placenta. Isn't that interesting? And then we have to say, well, is that bacteria toxic? Does it produce toxins? And we're going to show you, yes, that's probably the way it works. This is a thing was published in Dental Economics, the periosystemic link, and you can read this, it's in your handout. But I think the bottom line here, which is in red, it's the consensus of the medical and dental professions at this time that there is an association between the mouth and body. Not 2007, they're finally getting it. I think Weston Price died in 1930 or 29 or something like that. But anyway, the level of causality is still up for debate among some researchers and clinicians. This kind of comment's what led me to tell people or make the comment that I felt like I was in an 8-year argument with the town drunk with regard to certain things in dentistry. I mean, you know, you can always have a difference of opinion. I mean, me and my hunting dog disagree every now and then, but I think that that doesn't really prove much of anything. But I think we can now look at this and say, well, what's the research showing? And the point I'm making is that almost all the data I'm going to talk about now was published before this gentleman wrote this article. And this was in Oral Microbiology and Immunology, 2006. And they're not in the same order as you have them because I tried to concentrate them a little bit on heart versus the other diseases. And it says here, the present study shows that maintenance of inflammation may be enhanced by the presence of periodontopathic bacteria. And, you know, and again, they're looking at things in atherosclerosis. And what much of this research will show you, if you go to atherosclerotic plaques, the one thing you will almost invariably find there is a bacteria that came out of the mouth. And it's hard to you know, imagine swallowing that and getting into it. So it's going in through the blood, through the periodontal disease, or through some other problem. Again, they talk about bacterial diversis in aortic aneurysms determined by 16S ribosomal RNA gene analysis. That's where they use the PCR to amplify this gene and try and identify what bacteria is in the aortic aneurysm. And it says a wide variety of bacteria, including oral bacteria, was found to colonize aortic aneurysms and may play a role in their development. I don't think I have to explain to you what happens with an aneurysm if you have it in your aorta once the system gets weak. And the production of toxins at this site by these bacteria would prevent this cell from repairing it. I mean, this injury that might have started the aneurysm from developing itself. This other one here, serum antibody response to periodontal pathogens and herpes simplex virus in relation to classic risk factors of cardiovascular disease. Again, not done in the United States. And you can read it, but if we go down here, it says, the infectious burden comprising herpes simplex virus and periodontitis may increase the risk for cardiovascular disease by clearly decreasing HDL cholesterol concentrations. HDL is supposed to be the high density or the healthy cholesterol. And that, whether you want to believe that or not, I don't know, but it does point out that the bacteria are involved in this particular disease response. Periodontal disease in patients with ischemic coronary atherosclerosis at a university hospital, done again in Brazil, not in the United States. And if you look at this, it says periodontal disease was very prevalent in the group studied at higher degree of severity in those with ischemic heart diseases. The elevated prevalence of risk factors found indicates that intervention strategies are required. And I think this is something that's a good take-home lesson for people in dentistry. You have to attack the problem of periodontitis because you're not just saving the person's tooth, you're enhancing the health. You're enhancing this person's ability to live to reach the average age that most of us die. If they don't have that, they're going to have severe problems and they're going to die early and they're going to be a burden, you know, on the medical industry and and our insurance premiums and everything else. The bacterial profile and burden of periodontal infections in subjects with the diagnosis of acute coronary syndrome, again, not done in the United States. And the conclusion, it said, is it possible that stimulation of host response to oral infections may result in vascular damage and the inducement of blood clotting? In the conclusion of this, the oral bacterial load of S. intermedius, S. sanguis, Streptococcus aeruginosus, T. forensis, and T. dincola, and P. gingivalis may be commitment risk factors in the development of coronary heart disease. I think all the work we're looking at, I haven't yet read a paper where they didn't find this. So you have to say, well, when does something become so overwhelming and the data becomes so overwhelming that you begin to believe it? And again, this is 2005. Early carotid atherosclerosis in subjects with periodontal diseases. The present results indicate that periodontal disease is associated with the development of early atherosclerotic carotid lesions. The problem that you have when you're a scientist, if you look at something and you're proposing, say, that oral bacteria cause a problem, the one question you have to ask, what else could do it? And is there evidence that what else exists there? And that's what I would propose to you, and for you to propose to the people who think that periodontal disease and infected teeth aren't involved. What else does it? And what's your proof? There comes a time when you have to stop and say, there is nothing else that does this. What else could be causing this infection and the tissue breakdown at this site? So this one here is the evaluation of the incidence of periodontal associated bacteria and atherosclerotic plaque of coronary blood vessels. Very distinct. Again, not done in the United States. And what they were looking at, unstable atherosclerotic plaque is a dangerous clinical condition, of which I think we all know that. I don't know what they mean by unstable atherosclerotic plaque, but I think they're talking about plaque that breaks off and can clog arteries. And what they come to a conclusion, they come to several conclusions, it's in here, and it's kind of up in the air, but it says what is important is the presence of an acute inflammatory process expressed by significantly higher bleeding index in those patients with whom the examined bacteria species were found in atherosclerotic plaques. I would tell you, when you start breaking down tissue, the bacteria is present there. It's not an energetic thing going out. It's chemistry. Do the bacteria, pathogenic bacteria, release chemicals that cause the tissue to start breaking down? When the tissue and the cells start dying, the body responds with an inflammatory response reaction, rushing certain types of proteins called inflammatory proteins into that site, and later on we'll show you what those proteins are. This one involves periodontal disease and stroke risk, and they say the evidence on the association between periodontal pathogens and stroke is lacking. That means they haven't done the research. You won't get it funded probably by NIH. The conclusions. The present prospective study provides serological evidence that an infection caused by major periodontal pathogens is associated with future stroke. In other words, if you're putting toxic producing bacteria into your bloodstream, if they settle in the wrong places, you are susceptible to stroke. And I think all of you know how important stroke is to the elderly or people in this country. Here's one, endotoxemia, immune response to periodontal pathogens and systemic inflammation associated with incident cardiovascular disease events. Again, not done in the United States. And it says in periodontitis, overgrowth of gram-negative bacteria may cause endotoxemia and systemic inflammation leading to cardiovascular disease. And their conclusions, our results suggest that exposure to periodontal pathogens or endotoxin induces systemic inflammation leading to increased risk for cardiovascular disease. Now I put this on for one thing, because they talk about endotoxins. This is lipopolysaccharides, etc. What I'm going to show you, the most severe toxins produced by oral bacteria, anaerobic bacteria, are small molecules, small toxic molecules that can go wherever they want, and they pass into the hydrophobic aspects of the body and can cause very severe breakdown of the tissue. So while I believe in endotoxins, and I know what they are and I know they're very toxic, I think because you can buy antibodies, and most laboratories can measure this, they don't want to talk about hydrogen sulfide, methylthiol, and other compounds because they're a little more difficult to work with because they're so small. This one here is an evaluation of the incident of periodontitis associated with bacteria in the atherosclerotic plaque, again published in 2007, Journal of Periodontology, not done in the United States. In patients with a severe form of chronic periodontitis, it seems that clinical attachment loss is not associated with bacterial permeability into the coronary vesicles. What is important is the presence of an active inflammatory process expressed by a significantly higher bleeding index in those patients in whom the examined bacteria species were found. But I think the more you have toxic response around the capillaries that surround a tooth in the gum line, the more likely it is to bleed. Now this is, I think, one of the very important ones, and this is the correlation between atherosclerosis and periodontal Pewdie pathogenic bacteria in infections in coronary versus internal mammary arteries. And what they did is they looked at the effect and the infection level in the mammary arteries versus the coronary arteries. And the conclusion down to the absence of Pewdie pathogenic bacteria in internal mammary arteries, which are known to be affected rarely by atherosclerotic changes, and their presence at a high percentage of atherosclerotic coronaries supports the concepts that periodontal organisms are associated with the development and progression of atherosclerosis. In general, what they're saying is mammary arteries don't become infected with periodontal bacteria. Those in the coronary or in the cardiovascular system, or the I'm losing my word now, in the coronary arteries do, and that's where you get the atherosclerotic plaques built up. And the pressure in your blood system is more near the heart. If you look at it, most all the plaques that you'll see that are built up are where the heart pumps and you have a bend in the artery or something like that, and that's where you have the major blockages with the formation of atherosclerotic plaques. So it makes sense that the pressure of the heart beating as we age and with other problems we might have, if we form a break or a lesion in the artery, and when we have bacteria floating around the bloodstream from our mouth, they set up housekeeping in that site, and they prevent that site from repairing normally because of the toxins they produce. This is an antimicrobial prophylaxis in oral surgery and dental procedures. It talks about transient bacteria as a known risk factor for following oral surgery and invasive dental procedures in patients with altered immune system response and those with a susceptible site of infections, like patients with heart valve prosthesis or recent joint replacements. Essentially what they found in here, that anaerobic bacteria were found in 64% of the blood cultures. And they found it also in the plaques that they were looking at this time. And this is osteomyelitis with proliferative prosthesis in an unusual case. This was a single person. And they said, we present an unusual case of chronic osteomyelitis with proliferative prosthesis affecting the mandible of a 12-year-old patient. The source of the infection was related to the developing lower left third molar, which apparently had no communication with the oral cavity. And so I would wonder, where would the lower third molar get a bacterial infection if it had no communication with the oral cavity? How would the bacteria get into that and then get into the jawbone of this patient? And that's something that I think if any of the dentists have an answer for this, I'd sure like to hear it, because this was one that really confused me, a paper that I didn't understand. But it does point out that oral bacteria get into the mandible of certain patients and create a severe problem, which some of us might call cavitations or jawbone infections. Now we get into the part that I think is different than the heart disease, which is being studied quite a bit, but it's the evidence of pathogenic microorganisms in the placentas of women with preeclampsia. For those of you who don't know, this preeclampsia is a pregnancy-specific hypertensive disorder that often leads to maternal morbidity and mortality. It's not to be taken lightly. The conclusion of this study is the significant presence of parapathogenic organisms, microorganisms, or their products, and I would emphasize the products, in human placentas of women with preeclampsia may suggest a possible condition of parapathogenic bacteria to the pathogenesis of this syndrome. And I would go back and say, this is classic academic double talk. May suggest, and then sometimes they say may possibly suggest, or possibly may possibly suggest. There's a time when you say there's no reason you should have pathogenic bacteria, you know, in the placenta of a mother that's carrying a child. It just shouldn't be there. And it doesn't may suggest, it does suggest. As a matter of fact, some people would say it would prove that there is a definite problem with this person if it doesn't exist in the placenta of people who don't have this problem. Taking it a step further, the isolation of commensal bacteria from umbilical cord blood of healthy neonates born by cesarean section. Again, this was not done in the United States. And this study suggests that term fetuses are not completely sterile and that mother-to-child specific efflux of commensal bacteria may exist. They isolated bacteria from the breast milk of a healthy woman, E. facilium, and they injected this into pregnant mice, I think it's intraperitoneal, and the labeled stain could be isolated and polymerase chain reaction detected from the amniotic fluid of the inoculated animals. In other words, they could take a bacteria that went from a mother's mouth into her breast milk. They isolate that, they amplify it, they inject it into a mouse, and they find that when they inject it into the mice who are pregnant, that bacteria ends up in the baby mice, the ones that are in utero at that time. So these bacteria have the ability to spread throughout the body without any help. Even if they're not killing the animal, they can do it. It says, in contrast, it could not be detected in samples obtained from a non-inoculated control group. When people tell you bacteria from the mouth can't, by injection or getting into the bloodstream, affect certain organs or get into babies or into the uterus, etc., or into the cord blood, they're totally wrong. The research says they're wrong. This was a very complete study, and again, it's referenced here, and this was done in 2005. This is something that should be of interest to all the medical doctors in here. Is periodontal infection behind the failure of antibiotics to prevent coronary events? This was published in 2007, and in Atherosclerosis. And it's a kind of a complex study, but you can read it in detail, and you can read the rest of it. But it says, this paper presents the hypothesis that periodontitis is behind the failure of antibiotics to prevent coronary events. We discuss the systemic effects of periodontal infection and consider studies to test our hypothesis, which offers a novel viewpoint for the discussion of antibiotics in coronary disease prevention. I want to tell you how novel this is. If you read Weston Price back in the 1920s, he was saying this. I mean, there were things saying you cannot cure somebody of an infection, certain levels of infection, as long as you keep an infected tooth in that person's mouth. So this is hardly novel, but it's novel to people today, perhaps. And what they're saying is that no matter how much the antibiotics would prevent or enhance the killing of a bacteria that's in the heart tissue and perhaps save that person, if you are reinfecting that person with bacteria coming out of an infected dead tooth or severe periodontal disease where the antibiotic doesn't work because it doesn't get there, you're just beating a dead horse. You may help the person for a period of time, but if you don't tie medicine and dentistry together, and I mean join it at the hip so it can't be separated, you will not cure these people, not of the infections that cause these illnesses. This is one you can, I'm not going to go over this very much, but it just says the bacteria recovered from these infections are often of oral origin and involve mixed anaerobic oral flora. This is another one. I'm just going to skip this, but you can read it, and it just talks about the influence of an endotoxin on mice. But the experiments indicate that the pathogenicity of a mixed gingival flora may depend on a decrease of the local and systemic disease mechanisms induced by endotoxins derived by gram-negative bacteria. I want to tell you, I believe this, endotoxins would do this, but they are totally ignoring the fact that these pathogenic bacteria make chemical toxins that are relatively lethal. This is one that may be of interest to the mothers. The distribution of 10 periodontal bacteria in saliva samples from Japanese children and their mothers. And if we read this, it says our results indicate a correlation between presence of periodontal bacteria in children and their mothers, while the presence of red complex, this is the bad set of bacteria for periodontal disease, in children was highly associated with that of their mothers. In other words, mothers who kiss their children, who feed their children from the same spoon, are actually infecting their children with their oral microorganisms. And for those of you guys that like to make out with strange ladies, you're getting the same treatment. So the thing is that these infections do transfer, and there's something that you really need to take into account. So what we've come down to is, we need a study of the toxins associated with avital teeth and osteonecrotic materials. And if periodontal disease causes any systemic illness, then teeth harboring the causative anaerobic microbes should also contain potent toxins. And this is where I think that my research has come into play. And it's not novel with me. A long time ago they talked about, people with bad breath. If you have death breath, you have periodontal disease. You're producing hydrogen sulfide, methylthiol, putrescine, cadaverine, a lot of small chemical toxins. And I would absolutely tell you that the entire dental community, with maybe the exception of 0.01% of the researchers, are totally ignoring the effect of these toxins on human health. And I want to show you some of the results of what we've done. And I would like to point out that I started doing this because of the people sitting in this audience. There were a whole load of you that were talked about the problems that came along with this. And this is research that we did at my university, in my department, in my research lab to start with, and it's now done at ALT. And for those of you who send us toxic teeth and we send back and tell you, yeah, the tooth you extracted was toxic, this is how it's done. We take the tooth, we add it in 1 ml of sterile water, and we shake it for 1 hour. We remove that extract and we save it. We don't do anything with it. We then put it in another ml, shake it again for a second hour, for another hour, and we remove that. And we don't do anything with it except research. And then we do the third one. And when we get that third one and we come out, we take this third 1 ml extract. And for those of you who keep it in context, a ml is 1,000 microliters. So it's the 3,000 microliter wash in 3 sequential things, and that's what an analytical chemist does. 3 washes, gets off most of everything. And this says it's just not on the outside. At this time, it has to be coming through, and we'll show you the amount of protein that comes through and the amount of toxin that comes through. And this is where we do the work. We don't do it over here. If we did it over here, most of the teeth we tested would be toxic. With this one, we find out about 20 to 25% of the teeth don't display toxicity, so it allows us to put things on a scale of quarters. You know, very toxic, moderately toxic, slightly toxic, and not toxic. So what do we have now? You just saw what we did, and let's look at this. There's several in here. They're all underlined. If they're underlined, this is the number of washes. The first wash, the second wash, and the third wash. Put it in between. But if you go to something like this is the first wash, see how toxic, how much protein is out here, albumin, etc. If you go to the second wash, it decreases. You go to the third wash, it's minor. But there's still some proteins in there. And if we add this, and here's a really infected tooth, and keep this in mind, because when you do the topaz, if you're doing and you do the blue collar, you look at the protein level, you look at the level here versus the level over here, and you say which one of these teeth is the most toxic. And we'll show you in just a second how this tooth, the toxins in this tooth, totally inhibit the photolabeling of these 4 enzymes that we have here, and that washing it from first to the second to the third, we get the protein levels down dramatically. And this is early research. This is not what we do today. And we have a technology. There's no way I could explain this technology. It's called nucleotide photophonetic labeling. Suffice it to say that NIH funded me to develop this for 25 or more straight years. But if you look at the thing, let's take this really toxic tooth. This is the one that had the most protein. And I'll just go back a second. We're going to be talking about this one and the 3 washes that go along with it, and compare it to some of these others over here that have less protein. Here's the control protein. This is the one that you should be looking at saying, here's the level. If you go over here, that first wash, which is incredibly toxic, wipes it out. Even the second wash wipes out most of it. And the third wash does it less because we've diluted the toxins that are associated with that. But if you look at all the proteins in here, you can say we have an increase of protein, and when we have an increase of protein, we generally have a big increase of toxicity. And the concept of this is, as these bacteria that invade this site release toxins, your body fights it. It's similar to like when you get a spot of poison ivy. You get that liquid blister. That's because you're getting all these inflammatory proteins, such as serum albumin, rushing into that site. The water follows it. You get a swelling, but the proteins will bind the toxin and prevent it from going into your body and causing destruction elsewhere. And you can see that this one was really toxic and really wiped it out. Over here, where we had the less toxic tooth, there's the first wash, second wash, and third wash. You can see that while the protein is still there, it's toxic, because there's a decrease in the activity of this enzyme, because the lower the black, the less intense the black level, the more toxic, the more the proteins are in here. So later on, we'll show you. We did a study where we took out the proteins to see what was causing it. I want to point out one other thing. Look at this. This is bovine serum. This is the albumin level in humans. And this is where it runs in this thing. In the one that's really toxic, look at this albumin. It's running very abnormally. It doesn't run like regular albumin. And when we go to jawbone cavitational material, there are a lot of people that do jawbone osteonecrosis or cavitational surgery, and they send it in. Almost all the protein that's in those cavitations, and it looks like a mixture of motor oil and cottage cheese many times. But if you look at the proteins in there, it's almost all albumin, but it's albumin that's cross-linked to other albumin molecules, like it's been into a place where there's been a lot of very rigorous chemical activity. And plus, at the same time, that same material will be very high in mercury, silver, cadmium, iron, etc. I mean, so those jawbone cavitation materials are incredibly toxic. They're high in metals. They don't have normal cell proteins. They have proteins that look like they've been charbroiled. So we want to study the small molecule toxins associated with avital teeth and osteonecrotic materials. And it's important to remember that this site contains both small molecule toxins and proteins, and that we can separate these rather easily. And doing so proves that the toxins produced by oral anaerobes, not spelled correctly, are small molecules like hydrogen sulfide and methyl thiol. And so here's the second step we do. When you send your tooth in, we take that third wash and we place it, this is the third wash from the tooth extract that we just talked about, and we put it in what's called a centricon. There's a filter here that filters out all the proteins, and you put it in a centrifuge and you spin it down, and what you get is a protein-free ultrafiltrate of tooth extract containing just small molecules. Big molecules don't go through this. You can't find any protein in this material, and this is what we test. And we did this for several reasons, and one of them being that when we looked at the literature about pathogenic bacteria, we found out that the toxins in there were not proteins, not the endotoxins. The things that cause other cells to die were small molecules, or appeared to be small molecules. And so this is how we set up our test. We took all of these proteins, phosphoglycerate mutase, pyruvate kinase, phosphoglycerate kinase, creatine kinase, adenylate kinase, and acidic fibroblast growth factor. The reason these were selected, they all bound ATP, and they were all of a different molecular weight, so we could separate them on the gel and look at the effect of the toxins on all these proteins at one time. But on this one, we screened them, and we screened them by doing this. We did nothing, or we added hydrogen sulfide, which we know is very toxic at a very high level, 2 millimolar. That's how high it is in a periodontal pocket, by the way. And you can see it totally inhibited every one of these enzymes. Hydrogen sulfide is lethal. It'll kill you. If you go near an oil or a gas refinery, you'll see all these little stakes out there with a little building. They're measuring hydrogen sulfide because if it builds up, it'll kill the population. So they have warnings for this. It is an incredible toxin and is produced in your mouth, as we'll show later. And then this T is the toxin from a tooth. And so here's with nothing, here's with the tooth extract, and here's with hydrogen sulfide. So you can see these toxins are every bit as toxic as hydrogen sulfide. But I'm here to tell you they're not hydrogen sulfide. When we use this level of hydrogen sulfide, it smells like rotten eggs. If I were to open it up, a vial, a 2 millimolar, and set it here before my talk was over, everybody in the room would be complaining about the smell. These teeth, they have a smell. Trust me, they'll turn you green, but they don't smell like hydrogen sulfide. And some of them that are really toxic don't have much of a smell at all. So we're dealing with I'd say a compendium of chemical toxins that we don't totally understand. One of them that we know about is called gliotoxin, and it's at the end of my talk if I get there, but it doesn't smell, but it's more toxic than any of the rest of them put together, and it's produced by Candida. So what we can say is that the toxicity extracted from teeth are not large proteins, but rather small molecules. Also, this toxicity is mostly absorbed by charcoal, but not by cation or anion exchange resins. And what does this tell you? And I know a lot of you guys carry charcoal tablets when you go to Mexico. And when you start feeling a little sick, you take them. Why? Because why do they work? They soak up the toxins produced by the bacteria that have infected your intestinal tract. They also soak up your vitamins, so don't take them too long. But this tells you that these toxins penetrate, they're hydrophobic. Things that bind to charcoal, for more or less, are things that are very hydrophobic. In other words, they would penetrate the hydrophobic or fat tissues of your body very, very effectively. They would go through cell membranes, and they cause a lot of problems. And that is part of, we've talked about a compound I'm making, and this is part of the rationale behind getting something that would go in there and pick up these toxicities. And so what we've shown you here is that the presence of this type of toxicity with hydrogen sulfide and similar compounds can be detected in a radiodiagnostic assay using binding this. And this is nothing but hardcore science. I mean, nobody's ever, this irritates a lot of people, as you might imagine, but they haven't done this and said I'm wrong. And this has been now, what, 12 years or so? So here's the study as we do it today. We have these enzymes that we use that are abbreviated here, and you can see there are no other proteins in here because we filtered them out. So everything we're looking at in these studies is just nothing but small molecule soluble proteins. And we have two controls, and these are the proteins that we add. These are the enzymes that we add to the mixture. And then we can compare 3 root canals. Everything you see here, these are all root canals, and if they're in the same color, it's because they're from the same mouth. Like these 3 are all from the same person. And CA means cavitations, and we've got the control plus and a control minus, which is where we have to show that it's what we get without toxin and what we get when we add hydrogen sulfide. And when we evaluate this, what we can see, the two outside lanes are our control. You can see that these proteins interact and become radioactive very quickly. If you have root canals like these first three here, this one here is not nearly as toxic as the other two next to it. The two green ones, the outside one is incredibly toxic, and the one next to it's not toxic at all. And the person in the light color here, he had the least toxic of the root canals in his mouth. Now, why is that? The type of infection, and possibly this person who had the root canals that don't look like they're so infected probably practiced really good oral hygiene. Probably gargled with hydrogen peroxide and treated his mouth very carefully. Probably didn't eat a lot of sugar, etc. There's a lot of this, a lot of variability in how somebody's root canal and how much toxin it will produce. And we're doing an experiment now with one of my people that has a periodontal site, a very nicely located one that I can get into with a micro brush, and he can get into it and he tests it himself. And it's amazing how the level of toxin production changes with what he eats. It's just a one-person thing, but hopefully it'll bring up some interesting information. I mean, some days that periodontal pocket is incredibly toxic, and the next day it's not, depending on what he eats. We talk about cavitational materials. We have tested people who do jawbone cavitation. We've never found one that wasn't, reasonably toxic, if not incredibly toxic. And I would tell the people here that might get attacked on this. I represented or was an expert witness and helped a dentist that was being attacked for doing jawbone surgery. And we took 9 of his samples, and I did mass spectrometry on every protein that we separated out on a gel like this. Every protein we tested turned out to be human serum albumin, even if it were a higher molecular weight or a lower molecular weight, because you break it up into peptides by mass spectrometry. And what it says is that the material that you get out of these jawbones that are infected is where you've had a lot of inflammatory process. Albumin has been rushed into that area, and the chemistry in that jawbone, that place where you need jawbone surgery, is such that it totally disrupts, cross-links, and breaks down albumin protein. We could hardly find, we found a lot of blood clotting proteins in there also, but it was blood clotting proteins and albumin were the major proteins that we identified in the cavitational material from this person. So that eliminated the argument that the person from Temple Dental School was saying that he was removing normal tissue. It's not normal tissue. We also took that same tissue and did ICP mass spectrometry to identify the metals. And what you find when you do that, the metals aren't consistent with it being normal tissue either. It's high and heavy metals such as, you might imagine, mercury, iron, silver, copper, and other compounds similar to that. So the material that's in these jawbones that you guys that are doing the surgery, it's not normal tissue, and it's probably something that needs to be removed. And I think it's something that we really have to consider, and I think that maybe 12 years from now, if I'm still alive and here talking, this will be, again, something accepted, such as the focal infection theory. This is the bottom line here. We say osteonecrotic material do not have the metal content of normal tissues, nor do they have the normal proteins either. To back up the chemistry, and just to show you where a lot of the products come from, and some of the studies that we haven't done, when we look at the breakdown of L-cysteine, desulfhydrase, which is the enzyme that's in these anaerobic bacteria, and why they produce hydrogen sulfide, and why if you don't brush your mouth regularly, you start stinking. And we look at this, it produces hydrogen sulfide. It also produces ammonia. And what we're finding is that maybe the hydrogen sulfide plus the ammonia together are what make these sites so toxic and so susceptible to breakdown of the tissue. And this is what got me started. And this tooth was sent to me by Hal Huggins. There was a time in my life when I found Hal Huggins a major pain in the rear end. There's some things about him that I like and some things I don't like, but you can't talk about his intensity. And when he found out that I was visiting you guys and talking about mercury toxicity, he sent me some teeth, called me, wrote me, in general harassed the living daylights out of me until I said, we've got to do something for this guy so I can shut him up. Because I didn't think, as a matter of fact, I thought he was wrong. I mean, just to be blunt, I mean, I didn't believe that a tooth could be as toxic as mercury. And this tooth came from him, and it was extracted from a person with multiple sclerosis. And we did the same thing that we talked to you about. We washed his tooth 3 times, let it sit there, and at the, after the 3,000 microliters, we started adding microliters of this to this is a brain homogenate, by the way, labeled with ATP. And you can see that we go here. Here's what happened when we added 5 microliters of that last 1,000 microliter wash of a 3,000 microliter wash. 5, 10, 25, it just totally abolished the activity of the proteins in the brain. And this was something coming out of a well-washed tooth that had been extracted from a person who was sick. Now, I'm not saying periodontal disease or infected teeth causes multiple sclerosis. It might be that these people have a lower immune system and their teeth just become more infected. But what I can tell you is that a person with a tooth this toxic really needs some good dentistry and some good medical advice. And we need to be studying this area. There's absolutely no doubt that this tooth is more toxic than hydrogen sulfide. And it might be because of the ammonia that's in it also. It might be because of other toxins. And we don't have the answers. But we need to be pushing our government and the National Institutes of Dental Research to do more research in this area and not have people from Brazil, Chile, Japan, Sweden, Germany, and every place else do the research that we could probably do better and do more effectively in this country. But to say that these teeth do not have an effect on human health, whether this came after or before this person developed multiple sclerosis, it shouldn't be in their mouth. Not this level of toxicity. This is incredible toxicity. And, and I give full credit to Hal Huggins for pushing this issue. And I would also say Dr. George Mining, because coming to one of your talks, I didn't listen to Hal very much until I listened to Dr. George Mining give a talk on root canal cover-up. And when I listened to him, it was sensible science that he talked about. And sensible science is what wins this game. That's the reason I think the Academy today is winning this fight about dental amalgams. You do sensible science. Show me your sciences. Go back to Mike Ziff's mantra. So if we say, is there anything else that ties this together? And this is some of the research that most of you know brought me into this fight. And this is a polyacrylamide gel, and we've separated proteins after grinding up the brain of of a normal person and treating it. And we see what goes down in the pellet and what stays soluble. In normal brain, and that would be like over here on the side, here's a pellet and here's a supernatant, tubulin stays in the soluble aspect. And I would point out that in normal brain, tubulin, creatine kinase, and glutamine synthetase all stay soluble. If you do the same thing with an AD brain, for certain, tubulin and creatine kinase all go down in the pellet. It's an abnormal partitioning that anyone can observe. I've published it and people have said I'm right, but yet it's ignored. So what makes tubulin and creatine kinase, soluble proteins, go down in the pellet? And it's because they're abnormally affected by some toxicant in the brain that renders them not soluble in water, or renders them to bind to particulate material. If we take a soup and a pellet right here with zero treatment, and we look at the supernatant, there's the tubulin. As we increase the addition of the toxic material, that's 2.5 micromolar, 5, 10, and 20. That's very low levels. And as you go across here, you see when we get out to the very end, you can see it, well, right here it's gone. But if you go here, you can see that the tubulin is gone. It's gone down in the pellet. It's right here in this pellet fraction, and you can see that it's gone here. Here it's not affected. So hydrogen sulfide does not cause the aberrant biochemistry that we see in an AD brain, but an extract from an infected tooth does. And I think this is something that's very important. Even if you don't want to say it causes Alzheimer's disease, would you want something floating around in your bloodstream that caused your brain tubulin to abnormally partition? That was very hydrophobic, and the things that do cross into the blood-brain barrier, into the brain, are hydrophobic materials primarily if they're not supposed to be there. And if we look at the photolabeling activity, the hydrogen sulfide doesn't seem to have much effect on brain tubulin, but boy, this toxic material from this tooth with multiple sclerosis and other things totally abolishes the photolabeling and has no effect on this protein. And this is exactly what you see in Alzheimer's disease. It's exactly the same profile. So we can say that Alzheimer's disease is probably has something to do with chemical toxicities that are coming out of the oral cavity as well as anything else. So we have these results. But this is also observed in Alzheimer's disease brain and brain tissue exposed to mercury. This has been published a long time, and nobody has said I'm wrong about it. They just ignore it. This is the situation that's in your mouth as looked at a mouth by a chemist. And that's that you have amalgam fillings that release elemental as well as Hg2+. And you can have an vital tooth, it can be a different tooth, doesn't have to be the same tooth, that releases hydrogen sulfide and methylthiol plus other compounds. And when these interact with mercury, you get the HGS, and you might even get silver sulfide. But this precipitates out in the tissue, and this is what causes an amalgam tattoo. And if you don't believe me, excise some of it from the gum of one of your patients where it's purple, send it to doctor's data and have them measure the mercury content or the metal content. It'll be sulfides of the same heavy metals that are found in dental amalgams. And it builds up in the body. And it's probably not all that toxic to that tissue, although it looks kind of ugly. But if you look at this, if you make this from hydrogen sulfide, then from methionine you make this compound, and you end up making these two organic mercury compounds. We've made them in our laboratory, we've tested them, and we tested them by putting them in, and what we can tell you, they're incredibly toxic. And the toxicity of mercury compounds is based on their hydrophobic ability or their ability to penetrate cells. Hg2 plus is toxic to the kidney, but it's not too toxic to the central nervous system because it can't get in. But methyl mercury and dimethyl mercury are incredibly toxic to the central nervous system because they're hydrophobic and they partition into the fat layers or the fat tissues, and they penetrate the body, and they can get into places where we don't expect, and they are incredibly toxic. We can talk about the synergistic toxicity, and we can say this is a major problem, and we have to look at the ability and you know, getting amalgams out for no other reason than to prevent them from reacting to periodontal disease toxins, which we're not going to stop in total, is a good argument. You are manufacturing incredibly toxic organic mercury compounds in your own mouth if you have amalgams and periodontal disease. Mr. H.B. Wallace funded a lot of my research. He's now dead. He was the son of the Vice President of the United States under Truman. He was a person who thought that his father and other people suffered from mercury toxicity. And I would have to say that I steal a lot from people, a lot of it from some of you, but in this particular talk, Weston Price, I read all of his work, and I listened to Dr. George Mining talk, and of course Hal Huggins was the, you know, the tack that I had to set on to get this done. You can all tell him that too, by the way. And, you know, and I have a lot of friends in the IAOMT. A lot of you guys, I can't tell you how many times I've sat and talked with you and how much dentistry I've learned and the problems you have, and I really appreciate that. And like every husband, you know, I'm hard to put up with. I'm very intense, and, you know, I have a very tolerant wife.
I would like to present the Ziff Memorial Plaque to Dr. Boyd Haley. And in Michael's honor, I'd like to thank you for all the research. He really appreciated your research in the dental mercury issue. Thank you very much.